Hey everyone, I'm Dr. Fizz, and let's learn some physiology. All right, today, we're going to talk about senses. Senses are going to be super important for our understanding on this exam. I have no more things to say in my intro, so let's move on. All right, so we're going to cover a lot today, and hopefully this doesn't end up being a 25-minute video, but we're going to cover the reflex arc, muscle spindles, stretch reflex, Golgi tendon, withdrawal reflex, taste, smell, and hearing. Smell, taste, and hearing. All right, let's get into it. So time for the reflex arc. The reflex arc is something we've intuitively kind of been talking about this entire time when it comes to efferent and afferent pathways within our first unit. But we're going to go into depth a little bit more today and how they work within the spinal cord. So, long story short, the reflex arc works upon, for the most part, involuntary and voluntary movements. Involuntary or reflexive is, I think, is what we're going to be talking about today. Starts with a muscle rather than the brain. So we're going to be talking about how signals are sensed, go through the spinal cord, and then go back out to the system rather than having to go up to the central nervous or up the central nervous system. It'll mostly stay within the peripheral and go through the central to be able to make sure that it's doing the right thing. Let's see. So we'll basically go from muscle to spinal cord to muscle. And there are a couple of different things that are involved with our reflex arc. There are 5 basic components. The first one is, let me pull out my laser pointer. The first one is right here. We can see is a mechanoreceptor. Basically, receptors that are triggered upon touch or pressure or sound or motion. We'll talk a lot about those in a bit in this video. Another one is a chemoreceptor, which senses chemical stimuli, which we'll talk about in the nose or the olfactory system. For example, the lungs can sense O2 and CO2 and make decisions according to that, almost really in an involuntary way. We have thermoreceptors, which on our skin, which we talked about in our first unit, actually, and those relate to how our system responds to heat or cold. We have photoreceptors, which we'll talk about in the next video. Then we have nociceptors, which we will, I believe, talk about either in this video or the next video. I can't remember. We'll get to that. All right, moving on. So other parts that are important to that, I just covered the receptors. There are also afferent and sensory neurons that are involved. We have a control center. We need an efferent, an efferent, and a motor neuron, and we need an effector. So, talking about our effectors there, we have both a sensory neuron and an effector, actually. We'll talk about muscle spindles. So, muscle spindles are, well, first we'll talk about something called proprioceptors. So, proprioceptors are monitor the location of limbs, body movement, and strain of the musculoskeletal system. And a type of proprioceptor is called a muscle spindle. So these things right here, you can see on the muscle, monitor muscle length, and they're bundles of modified skeletal muscle fibers called intrafusal fibers, which can run perpendicular to the extrafusal fibers that help contract. So they kind of go this way, and instead of this way. So that's kind of cool. Here we can see alpha afferent motors, so motor neurons that are going into the cell. And these will be gamma afferent motors, which are a different system, along with our intrafusal fibers. So there are two different types of muscle spindles. Those are nuclear bag and nuclear chain. Nuclear bag are larger and have 2 different nuclei clumped together, causing the fiber to bulge. So these are also called 1A fibers, type 1A fibers. We also have nuclear chain muscle spindles, which are nuclei in a row on the outer edges, kind of like the extrafusal fiber muscle nuclei that we found on our muscle cells from our last video. These are called type 2 muscles. So they're innervated by these type 1A and type 2 muscles. Type 1A is the largest diameter neuron and is myelinated, and it conducts pulses super rapidly. Type 2 is a little smaller, but it's also myelinated and it's in a chain. Gamma motors are two different types. You can see here that we have a gamma afferent motor. You have dynamic gamma, which is associated with our bag fiber, our movement, and you have static gamma, which is coordinated with our chain fiber innervation. So, for example, the thing I like to use is my bicep because it is massive, and so we will be using in our dynamic right here. Well, I guess right here is type 1A, type 1A, type 1A, type 1A, or bag chain, or bag fibers. And as soon as we get to here, the stretch, and we don't move, type 2, which is a chain, is activated. So that's important to remember. Type 1A, type 1A, type 1A, type 1A, type 2. And type 2 measures the length of time the bag or 1A responds to the rate of the stretch. So you could do this all day. You could do this all day long. You go super fast. That's 1A. And the moment it stops, it more measures how long it takes to get there and regulates how long it takes to get there without breaking the muscle. That's type 2. All right, so moving on from muscle spindles, we have a stretch reflex. So the stretch reflex is super important. It kind of shows us what muscles are in play in order to keep everything stable, or else we would break our own bones, surprisingly. So we have 5 different stages here that I want to show you. You can't really see 3 because it's so small in there. But step 1 is when we take that hammer and hit the tendon right there, which will cause our tendon to stretch. Or if we hit the tendon, it'll cause our muscle, our quadricep, to stretch, triggering on that alpha motor a signal that we're stretching the muscle. It'll go into the dorsal fiber, synapse with a neuron going up to the brain, signaling, hey, this is going on. But we won't even really have to send anything back because our system will take care of it. Stretch reflex. It'll send 2 different signals. One that's an EPSP, which stimulates the contractory muscle of the quadricep to try to bring it back to normal. Because if it overstretches, then it's going to freak out. So it wants to bring it back to normal. It'll contract. Another route there in the stretch reflex is that it'll go through something called an interneuron, and an interneuron will actually flip the signal coming in. So it'll be a stimulating signal, an EPSP, but it'll flip it into an IPSP to the antagonist muscle, which is the muscle that counteracts the muscle being activated right now. So it'll send that dotted line out to the antagonist muscle, which is our hamstring, in order to inhibit that and allow that to relax more. That way our quadricep can make sure that it contracts and doesn't get overstretched. Super awesome. In a nutshell, that's the stretch reflex. Basically, there's 2 different pathways there. One's an EPSP, one's an IPSP, flipped from an EPSP to our antagonist. Super awesome. Next. So, talking about our Golgi tendon, which is basically what measures tension, I should say. So muscle spindles sense stretch, and Golgi tendons monitor tension, and that's different. You can have something stretch and it's still not tense, like Laffy Taffy. Or I guess taffy in a shop, it can be drooping and you can still stretch it and it's not super tense. But a rubber band, once you get past a certain point, you can stretch it and it's going to stay pretty tense. That's different. Man, stretch and tension are related, but they're not the same thing. So the Golgi tendon is where stretch, where tension is involved, and it is inhibitory as opposed to the excitatory actions of the muscle spindle. So the Golgi causes relaxation and it prevents excessive tension in the entirety of the muscle's usage. So going through these points here, we have number 1 where there's a tension of some sort exerted on this tendon right here. The Golgi tendon will sense this tension and then send a signal on this blue pathway right here. A sensory neuron will conduct the action potential in the back. There's a lot of stuff going on here. We might have to zoom into that. Number 3 in the back here, sensory neurons will synapse with the interneuron. So there's 2 different interneurons happening here, also one to the brain. Both will be EPSP at first, but you'll have one interneuron that goes to the muscle being stretched there, or the tendon being stretched there, and it will cause it to create an IPSP. So it'll inhibit that. The alpha motor neuron to the quads will inhibit, and the alpha motor to the hamstring will be stimulated. So this is what allows us to move our leg back and forth, is the tension on our muscles. So if we were to maintain tension on our quad while trying to contract our muscle and our hamstring, that's going to be kind of difficult. We need to be able to relax our quadricep at the same time as trying to contract our hamstring. So, all right, and number 5 in there, I don't know if I wrote down notes for that, but basically sends a signal there, stimulating the contractory state of this. So, all right, moving on. So, talking about the withdrawal reflex, this might be a 20-minute video. Talking about the withdrawal reflex, we have our legs here. This guy's been hitting leg day, but not glute day, apparently. I didn't draw that very well. This dude has a sensory neuron, but you can see this little tack down here that he's stepping on. So he steps on a tack and he breaks his mama's back, and neurons are hit. It gets sent up to the spinal cord, says, ouchie, I've been hit. And synapses with a bunch of different sides, it actually ends up, without even us having to think about it, we're able to back up, pull our foot off, and then we can see what's going on. This is actually a reflex. It's pretty cool. It'll synapse and send signals to both legs through interneurons, one being inhibitory and one being excitatory. So if you can imagine, you step on a tack and your right leg will go up. So what contracts your leg upwards? It would be your flexors, which is in your hamstring. So your alpha motor to your hamstring will be an EPSP, whereas to your quad, you don't want to extend that guy to stab yourself more with the tack. It'll relax. And then in order to stabilize yourself, it'll go to the other side of your spinal cord. The interneurons will, and they'll send a message that you need to relax your flexors and contract your extensors in order to maintain stability on that foot and balance on that foot while you raise your other foot. Pretty cool. This is called the withdrawal reflex, and it happens when you touch a stove. Your hand always will go back towards you. Your flexors will always contract, and the flexor that's being on the limb that's being hit will always contract, and your extensor will always relax. And then if you need to balance yourself with something else, it'll be the opposite. So, okay, moving on. Flying. So, 14 minutes. So, all right, smell. There, let's see, we have a nose here. This is talking about our sense of smell. So we have something called an olfactory receptor. And these are just neurons. They have a lifespan of about 2 months, and they're pretty small. They're about 100 about a 10 square centimeters of area, so not very big. Dogs, on the other hand, have like 100 square or like 1,000 square centimeters of area, I think, in their nose, and that's why they can smell super well. It's either 100 or 1,000. Don't quote me on that. So, going through this, at the top of this we have receptors over here that'll pick up on signals, and we'll talk about what actually happens in these receptors. These receptors specifically are called olfactory neurons. Going up the chain, and they reside on what's called an olfactory epithelium, which is number 4 right here. They go through number 3, which is just bone. It protects it. It protects the olfactory bulb, which is number 1. These neurons will then synapse on the soma of these mitral cells, which go up to the olfactory receptor in the brain. And number 5 right here, this little area where everything comes together, is called the glomerulus. I had to write that down. I didn't know how to say that. So that's what happens. Let's talk about what actually happens on a molecular level. On a molecular level, we have a little bead over here that this guy comes in and well, side note, you can actually smell 1.7 trillion different smells, and each of them have to do with little particles. So a little particle will hit a specific receptor, and that'll trigger a G protein, which will releases alpha neuron, which will hit something called adenylyl cyclase. Adenylyl cyclase will release CAMP, which is a cyclic AMP, which is a messenger protein. Messenger protein, I believe. The messenger protein here, cyclic AMP, will hit our channel that allows sodium and calcium into the cell, which will depolarize our cell. That depolarization will cause our voltage-gated chlorine channels to open up and allow chlorine to leave. And this will even further depolarize the cell, because negative particles will be leaving, and so that'll cause an action potential up to the neuron that we saw. Alrighty, moving on. Let's talk about taste. So this one's a little ghetto. This drawing, these are from my class notes. We have 4 different types of something called papillae, or the sensor, like the buds on our tongue. These 4 are called circumulate, the fungiform, foliate, and thyrliform. You don't really need to know those, but long story short, you have grooves in your tongue, and all of these papillae hold different types of taste buds. These little fan shapes right here are actually the taste buds, and they reside all throughout here. So these little taste buds allow for different senses to be received. You have 125 different cells, but really we only care about one type of cell in your taste bud, and that is called an epithelial cell. So this epithelial cell can receive different signals, and depending on what happens within this cell, based on what you eat, you'll sense different things. So we can sense salts. We can sense salt because we taste positively charged ions, like sodium or potassium, I guess. We also taste sour, things that are acidic, or if you've taken OChem, hydrogen ions make something acidic. And Gen Chem, I guess, is a Gen Chem thing too. So we also taste sweet, which is a carbohydrate. We taste carbohydrates through these taste buds. We taste bitter things, which is Quiznos. I don't know how to say that. Quiznos subs. And then you also taste a new thing that they've discovered recently is called umami, which is a specific type of taste, and usually glutamate is involved with umami. All right, so going through this, we have a taste receptor cell, or the epithelial cell. All of what happens from this dotted line and down is the exact same. You have a voltage-gated calcium channel, and you have a smooth endoplasmic reticulum. It just depends on which one is used, and we'll talk about that. So these three different subsections of cell reactions can be split into salt, sour, and then sweet, bitter, and umami are put in together. So with salt, you taste sodium, and your epithelial sodium channels will actually open, allowing allowing sodium to go through. And here, give me 1 second. Allowing sodium to go through, and it causes a depolarization within the cell. This depolarization will allow the opening of this calcium channel right here, which will clip the clip the snare proteins and allow them to attach to the end of the cell. And usually there's an axon right here, and that'll cause glutamate to be released and go into the cell. I'll go and fire a potential. And then you taste salt at that point. You have sour cells, sour receptor basically, where if you taste a bunch of hydrogen, hydrogen will be on the outside and the inside of this. It'll cause a depolarization. These potassium leak channels will actually be closed, the amount of hydrogen on the outside of the cell, and that will cause this depolarization will cause calcium to leak in and send signals as well. You have sweet, bitter, and umami. Sweet, bitter, and umami allow for the receptors to be hit on this G protein. It sends an alpha signal to the phospholipase C, and then IP3 opens intracellular calcium channels on the smooth endoplasmic reticulum, or the sarcoplasmic reticulum. I don't know for sure. Anyway, calcium channels are opened, and a G protein receptor lets them through. That allows for these things to be clipped, their snare proteins, and attached, and they send a signal as well. All right, talking about sound real quick. You hear something that goes through and waves through your inner ear or outer ear, sorry, and it waves your tympanic membrane, goes through your middle ear, and wiggles. Forgot what this is called, back and forth. It actually hits a membrane that is connected to your inner ear, where your cochlea resides. And let's talk more about the cochlea there. That's going to be important. So in the cochlea, you have something called a basilar membrane, and then nerves that receive signals and send messages out to tell the brain what you're hearing. It's super important, and that's why people can get pretty bad ear problems, is because this thing is actually messed up. So this is the inside of the cochlea. It's actually it's a spiral looking snail shell kind of thing, but this is, if you were to slice it in half, this is what the tube would look like. You have something called striae vascularis, which are blood vessels that go along the outside right here and don't really touch anything like the tectoral membrane, which vibrates a lot. If you were to have it over here, they would sound like rushing water all the time, and you wouldn't be able to hear anything, and it'd be super loud and it'd be really annoying. So our creator put this here in order for us not to have to do that. It's pretty cool. All right, so our tectoral membrane resides on top of these hairs, these hair follicles right here. There's one right here, and there's usually three right here. These send signals back to this, which sends a lot of signals back to the brain. Before we talk about that, though, we'll talk about, this is the endolymph. What happens on the outside of the cell is that something called NKCC1 will allow chloride, potassium, and sodium into the cell, and thanks to our sodium potassium pump that's pumping sodium and potassium in and out of the cell, this will cause a depolarization and allow calcium to leave, or chlorine to leave our channels. Potassium leak channels on the inside of this membrane will allow potassium in, which will help our sodium potassium pump that exists here, and then causing the same thing to happen on this side. It basically creates a barrier So we have NKCC on both sides, a sodium-potassium pump on both sides. It kind of just creates a barrier that allows potassium to flow through. Potassium flows through and creates about 150 millimole per liter solution on the inside of our cochlea there. And that's where the hair follicles come in. Hair follicles will kind of move like this in term when they're hit with sound, and you'll have a hair basically that'll poke into the end of each striation here, or this each I guess protrusion, and when they pop out, if the balance of potassium is right, and that'll allow potassium to enter into the cell. Potassium will enter into the cell and create an action potential, which will open up voltage gating calcium channels right here, allowing for the snare proteins on these vesicles to be released and sending a signal to the brain through this. Sodium potassium pump also keeps the membrane potential correct. If there are two different, I'm not sure what they're called, but something's here that basically help keep the membrane potential of this basilar membrane. But if they were to be ruptured, potassium could actually leak out of this area and cause a whistling, ringing sound, and that actually happens a lot with the older you get. So it's something you get to look forward to, unless you're hunting Shaw and you are already old. All right, so this has been my video today. We talked about the reflex arc, spindle, muscle spindles, stretch reflex, Golgi tendon, withdrawal reflex, smell, taste, and hearing. If you have any questions about that, good luck trying to get a hold of me. And yeah, this has been Dr. Fizz. Stay busy.